Hi, everybody. I'm Amber Mosley, and I'm here with Emma.
Hi, everyone. This is Emma Dowd.
And we're going to talk to you today about SEQUEST HT. So this is the main database search algorithm that we use for the majority of the proteomics core projects. And so we're going to give you a little bit of an overview of how the search algorithm works, and then we're going to tell you about some of the key settings and basically how we make a decision on how to set those settings. And so basically, the idea of a database search algorithm is that we use the FASTA database that we design. And in the FASTA database section, I did not mention that, but I will mention it here, that we also include sequences for a number of contaminant proteins that we know sometimes are in the sample. And so that includes things like trypsin, which we add to do the protease digestion, and also antibodies, which a lot of people use for immunoprecipitation. And so if we just do this performance on a desktop computer, and the reason that we do the analysis on a desktop computer is because this software is commercial. It's commercialized, and so it's fairly expensive to be able to get a license. And so we use it on a desktop computer, although we are able to use multiple cores. And so we have workstation computers that have a large amount of RAM and a good amount of processing power. And so this is just an example of one of our analysis computers. And basically what Proteome Discoverer does is it takes a database, and it first takes the mass of the precursor ion. So this would be the MS1 mass. And then it makes a list, depending on your precursor mass tolerance, of the number of peptides that are within that precursor mass tolerance range. And so the tighter or the smaller your precursor mass tolerance is, the shorter this list is going to be. And so it's going to take less time to do the computational analysis. And that's one of the advantages of high resolution mass spectrometers like the Orbitraps that we have in the proteomics core, is that they really help us to reduce the search time, because this list is way shorter when we're doing matches against the database to look at theoretical matches. And so when we do this, we have an experimental fragment match spectra, and this is showing from an MS2 or MS to the second experiment, or MS-MS. And so this is what the data would actually look like. And what SEQUEST does is it takes our entire FASTA database, and it's going to digest it in silico based on the settings that we are going to talk about in a little bit. So one of those includes the number of miscleavages that you can have from your protease. So for instance, if you're going to use the protease trypsin, if you look at this peptide here, number 2, in reality, this should be a peptide that is cleaved into 2 pieces by trypsin. You'll see there's a lysine residue right in the middle of the peptide, whereas this top matching peptide only has an arginine at the extreme C terminus, and this looks more like a tryptic peptide. So however many you allow, though, for this to be able to have this tolerance, you can get longer and longer peptides. That's one thing that can happen. But you can also have a different composition of peptides. You may have ones that are more basic in nature, because you may have internal basic residues if you have more miscleavages. And so that's something to consider. And in some applications, this is particularly important. For instance, if you're doing something like you're mapping modifications by an enzyme that modifies lysine residues, like an acetyltransferase or a methyltransferase, then you may have a situation where trypsin cleavage might be inhibited by the modification. And so you may want to increase the ability, the number of miscleavages that you can have because of your experimental setup or because of the amino acid sequence of your protein. This is something I can talk about all day. I'll try not to go on too big of a tangent on it. But basically in SEQUEST, you get these in silico digests based on the settings that we put in. And then you generate a theoretical set of MS-MS spectra. They're going to match to these particular theoretical peptides that are close to the M/Z, or the mass to charge ratio of our MS1. And so we take our MS1 data, then we take the associated MS2 data, and we compare our experimental to our theoretical. And then once that's done, SEQUEST uses a score that's called an X score, which is a cross correlation score. And put simply, this is just matching up how many of the peaks overlap within, again, a particular tolerance window. So we still have mass tolerance for the MS2 data. And I'm focusing on this because we're going to talk about this a little bit in the settings, why that's so important. And so how close this is between theoretical and experimental is really important. And the more accurate the mass spectrometer that you have, the closer that you're going to be able to set that tolerance so that you have a really good filtering mechanism to be able to have a high quality identification. So then we have a rank here, it's ranked by X score. And then you're also going to get another score that we'll talk about that's called the delta CN that says how far apart the first and the second peptide match are. And so you can see in this particular one, this X score has a cross correlation score of 6.5. And then if you look at the second best peptide, you'll actually see there's a pretty big drop. And so you go from 6 down to this 3 range. And so that's usually, this would be a very strong identification. And this is from a real data set. We then perform a false discovery rate calculation, which we'll talk about a little bit outside of this. The false discovery rate calculation is not done within SEQUEST. This is done with some other analysis pipelines. And then we're going to get our list of peptide matches that pass all of this criteria, as well as a false discovery rate calculation. We typically keep our false discovery rate calculations at less than 1% at the peptide level, right? And so we have peptide level false discovery rate, and then we also can calculate a protein level false discovery rate. But I'm not going to talk about that anymore in this portion. And so there are a lot of different computational approaches that can be used. And so SEQUEST, as I talked about actually in the FASTA database presentation, is one of the first, it's actually the first, the first algorithm that was developed for protein database searching for FASTA databases. And so it takes advantage of proteome FASTA databases that we can generate from NCBI or from Uniprot, and then the proteomics core we use mostly Uniprot databases at this point for reasons that I covered in the FASTA presentation. Soon after SEQUEST, Mascot was also published. I think that was in 1999. These are both commercial softwares, so you have to pay to use either one of them. Xtandem is an open source software platform, which also has some really nice functionalities. And then the PEAKS software package, which is a de novo sequencing that is database assisted, but does not require a protein database, is something that Emma will also talk about in another presentation. And Emma, if you've got any comments, just feel free to jump in. And so these are a lot of the different platforms where these different algorithms have been integrated along with visualization tools, false discovery calculators, and different things that help and aid in different quantitative pipelines. And so there are quite a lot of them. Some of you may have heard of like the proteomic pipeline. And so that's a fairly common one. And also MaxQuant, which just uses the Andromeda search engine. And so MaxQuant is also publicly available. And so they all have their different positives and negatives. And so a lot of them are used for different approaches. And of course, this is only expanding, like you see in a lot of different bioinformatics applications. And so we're going to talk about SEQUEST in this lecture. We're going to focus in on it, which was first published by the Yates Lab in 1994. And so it basically uses peptide databases and then this comparison of amino acid sequences by looking at the fragment ion spectra data and comparing that to the precursor mass so that you can look at basically what the picture is on top of the puzzle box. If you think of it from that analogy, the MS data is like all the pieces of the puzzle that you put together to make the picture that's on the front of the puzzle box. So MS1 and MS2 work together to be able to solve that information of this sequence. And so in addition, you get cross-correlation analysis and then outputs. There were a lot of different ways that this data can be viewed, but now these are really incorporated into a lot of these different pipelines where you can then view the data in a lot of different ways and also have a lot of nice visualization. And so SEQUEST has actually had a lot of development and updating since 1994 when it was first published. And so there are a lot of different iterations of SEQUEST, and it has also led to the development of similar but slightly different algorithms as well. So you can just kind of see that here. And so the one that we are using is SEQUEST HT. And SEQUEST HT is one that's allowed to do multi-threading. So there's a scheduler so that you're able to run multiple jobs and then pull that data back together. And so it's a way to use multi-core CPUs. And so it's a way to make things a lot faster, especially when you're using a multi-core computer. And so, but we can't put it, this cannot be put on a cluster. There are other versions that you can put onto clusters. And so it benefits from some other algorithms as well. And it also allows for handling of different types of fragmentation data, including electron transfer dissociation, which is a approach that we have on the Lumos mass spectrometer, and higher collision dissociation, which is actually employed on all of the mass spectrometers that we have in the core right now. So those are both different options. In addition to collision-induced dissociation, which it was designed for originally in SEQUEST in 1994. And so the required input, this is what our Proteome Discoverer-based interface looks like. And the viewer for this is actually open source, so you're able to use it as a viewer and look at the data. But to do the analysis, you have to have the licensing available, which we do have in the proteomics core and also in my lab. And so the required inputs are, you have the raw data, so we have the raw file format that goes into Proteome Discoverer, and then you define different parameters about the data that's in that, that file and how it was acquired. And then you're going to have your FASTA database, which is listed right here in the input name. There's the protein database. And then you're going to set a number of different instrument and method tolerances. And we're going to talk in more depth about some of those. And also a little bit about post-translational modifications and different peptide modifications that we work with. And then finally, another thing is you want to think about the number of mixed cleavage sites. So I talked about this a little bit earlier, but this is certainly enzyme strategy dependent. So if you're using an enzyme that's not very efficient, like let's say chymotrypsin, you may have more miscleavage sites than if you're using a highly efficient enzyme, like some of the engineered trypsins, like trypsin gold from Promega that we use in the lab, which is really highly efficient, and we get a few miscleavage sites. You can also do quick digestions with trypsin to be able to get low efficiency digest to have more miscleavages. That's a strategy that's used for analysis of some types of modifications, and to also generate larger peptides, which are sometimes optimal for ETD type fragmentations, and they can give you higher protein coverage, sequence coverage. And so there are a number of different strategies that can be used. But all of those strategies have to be integrated into your setup of the SEQUEST parameters. So in the modules over here, so each one of these modules, they're all from Proteome Discoverer. Percolator was also developed from an academic group. But this is just the settings for SEQUEST, and they actually go down a little bit further as well. And so there's quite a lot in there that we have to consider and match that up with the analytical settings. So the key settings are really mass tolerance. And so I talked about mass tolerance on that little workflow slide in the beginning, just because it's so important. And so precursor mass tolerance would be your MS1 tolerance, which is going to, again, in SEQUEST, define how long your list of potential peptides for matching is going to be. And so this one can make a really big difference on how fast your search will go. So for Orbitrap instruments with resolution settings around 45,000 or higher, the precursor mass tolerance is set at 10 ppm. You'll see some other some publications that will set this higher. I've seen it as high as 30 ppm, and then they may also have some slight adjustments on the resolution being a little bit lower. But this is in the proteomics core. We rarely go under 45,000, and so we set our precursor mass tolerances at 10 ppm. So in reality, what we've seen is that many of the spectra that we identify often have precursor mass errors that are under 1 ppm. We actually have some that are even like 0.1 ppm, but the tolerance is something that is a little bit fluid, and it can drift as you do experiments. So the longer that you go without calibration, or if you develop some burn or some ion burn in the instrument, you get a little bit, we'll call that dirt, but it's ion dirt. It's not like dirt from outside. So if you get the instrument is a little bit dirty, then that is something that you can see more drift in the ppm, the accuracy of the instrument. But the Lumos instrument and the Eclipse, which is the new instrument that we've recently acquired, we have internal calibrants on both those instruments that are leaked into the ion source, and they allow us to do some real-time correction of mass error. And they also let us know if the instrument is out of calibration, so we can just walk up to the instrument or remote into the instrument and look and see how the internal calibrant is looking and to see if it's a little bit off. And we get, I think, 4 or 5 decimal points to be able to look at that, so we can tell pretty quickly if the calibration is coming out. And so then this is one of the reasons that we calibrate the instruments weekly. And so we make sure that we have mass calibration once a week to try to prevent this drift from occurring. If we look then to fragment mass tolerance, the story is a little bit different. So we have a lot more variability in our acquisition strategies. And so the setting of fragment mass tolerance is fully dependent on the acquisition settings that we use in the mass spectrometer. So for any experiments where we're doing peptide sequencing in the ion trap, and so that would only be relevant for the Lumos and the Eclipse instruments, because they're the only instruments that have ion traps. When you do MS2 analysis there, the fragment mass tolerance is set somewhere between 0.5 to 1 Dalton, because that's a lower mass accuracy mass analyzer. So that includes peptide sequencing. When we do it in an MS3 based method, a lot of times in the MS3 based method, the peptide sequencing is still done in the ion trap. And so the MS to the third is just done for quantitative reasons. So depending on your instrument setup, this can be really important. You have to know exactly where your peptide sequencing is occurring, and then in what area of the mass spectrometer it's occurring, so that you set this tolerance up correctly. In addition, for MS2 analysis on our standard instruments for settings, even of like 15,000 resolution for MS2 for the fragment mass tolerance, which is used sometimes on the Q-Exactive instrument, you want to set the fragment mass tolerance to around 0.02 to 0.04 Dalton. And so that's going to be set right in this area. So there's a whole tolerance section, and it's right after you defining the enzyme and then what database that you're being used. So if you're doing a cross-linking analysis, which we have a lot of different acquisition strategies for cross-linking analysis, and so you really are going to have to look specifically at the cross-linking strategy that's being used. But if you're using an MS3-based strategy, in which the MS2 step is where you get your peptide sequencing, like in an ETD strategy, then you're going to base that on the MS2 settings. So you're going to use the ion trap settings for MS2 and the ion trap if you're using something like ETD. But if you're moving to the MS to the third, and then the MS to the second step, that's where you break the crosslinker, then the peptide sequencing is going to occur in the MS to the third step. And then you're going to want an Orbitrap based tolerance. And so it's just really important to know what the acquisition strategy is. And one of the reasons that in mass spectrometry data analysis and bioinformatics, it's always been heavily tied to the individuals that are also doing the data acquisition, because the settings in the data acquisition really, really help to set exactly what these need to be in the data analysis steps. And if they're not done correctly, this can lead to higher false discovery rates in some cases, or in some cases you just don't even see some of the data that you should be getting. And so there are a lot of caveats to this, and you want it to be optimized so that you get the most identifications that you're able to. So in addition, if we talk about modifications, so we'll see in here, down at the bottom, it's cut off. There are static modifications. There are also dynamic modifications. And then when we think about protein chemistry, you have modifications that are going to occur on the protein side chains. And then you have modifications going to be occurring on the termini of the individual peptides after cleavage or digestion. And then you have modifications that are occurring on the protein terminus, because there's a biological chemistry that occurs that's going to have modifications to the protein termini. And so you have to define dynamic and static settings for all of those different areas of the protein, so that the search algorithm knows exactly which type that you're looking at. And so if you're looking at heavily modified peptides, you also need to consider changing this max equal modifications per peptide. especially if you're looking at something that can be heavily modified by something like, we're going to use phosphorylation as the example here. So if you're going to do phosphorylation and this dynamic modification number 2 here after oxidation, we would add the phosphorylation, which is a plus 80. Dalton, it's actually 79 point blah, 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 but it rounds up to 80, very close, on serine, threonine, and tyrosine. So those are the amino acids that we're going to look at. And so if you're looking at max equal modifications per peptide, that is going to be on all 3 amino acids. So that's something that you have to consider as well, because it's not per amino acid per peptide, it's equal modification on a peptide. So you'll only have 3. And I'm going to show you that this actually, you can actually run that. You can have options where you can get more phosphorylation than that pretty quick in acidic regions of proteins. And then in addition, if you do need to set this differently, you can also adjust the setting, and you can perform side-by-side analysis, and then you can combine the data together in the percolator step. And we're going to talk about that a little bit more on the next few slides as well. So then, so one of the things is that SEQUEST searching does have limitations on the number of PTMs that you can search for at the same time. So there's only 6 options for dynamic modifications on amino acid side chains. And we want to have oxidation as one option, because this is a non-biological modification that happens when we digest proteins and they're exposed to molecular oxygen, that we have some methionines that get oxidized and others don't. And so we need to always include this as a dynamic search. But if you're looking for phosphorylation, acetylation, ubiquitination, methylation, and methylation comes in mono, di, and tri, you'll see you're already out of modifications. You can't search for all of those at once. And so to be able to do this in Proteome Discoverer, you can actually make parallel searches. And once you have 2 SEQUEST nodes here, and you're going to do 1 set of modification searches in this search and 1 modification search in this one. Let's say we do phosphorylation over here, and we do the 3 different types of methylation in this search, and then we're going to combine them all together at the bottom. That will at least let you cover that sequence space, but the problem is it will not let you assess whether or not you have both phosphorylation and methylation at the same time on the same peptides. And so you have to really be careful about what combinations that you used for your computational analysis and what the goals of your study are. So this is a little bit challenging to set up because of these different limitations. And this is not unique to SEQUEST. This is something that you see in a lot of the search algorithms, and it's partly because of the computational time required and the increases that you see every single time that you add a modification. And so sometimes you would generate a search that's so complicated that it would not finish in any reasonable amount of time. And so that's something that obviously you want to avoid. And so in the scoring step, which is also done in SEQUEST, you also have scoring done by false discovery rate validators, which we're not going to talk about in this section. So there are a lot of different options. But the search engine outputs are going to include X-score. So that's that cross correlation score. And we talked about that a little bit already. So I don't think I need to rehash this. And so there's X-score values above 2 are usually pretty good. But you'll see X-score values that are even, I've seen as high as 7 or 8. And so it really depends on the charge state. And then not the charge state as much as the amino acid length. It does depend on charge state, but charge state also depends on the size of the peptide. So these all kind of go hand in hand. So really big peptides are going to have higher X scores. There is a bias in the scoring for big peptides. So if you have a small peptide, it's going to have a relatively low X score value, but that might be okay because then that might be as high as it's able to go with this scoring metric. In addition, for delta CN values, this is going to tell you how different the first identification is from the second identification in your list. And so overall, the more separated those are, that's considered to be better. However, in post-translational modification searches, the delta CN values can be artificially high. And that's because you may not have clear fragment ion coverage that's going to give you site localization. And that happens also sometimes when you have a peptide population where you have a mixture of peptides which are exactly the same peptide from a chemical perspective, but the modification is on different amino acids, which has really big impacts on the biology. So this is a very important distinction to be able to make. And I'll show you an example of that. This is a peptide from this protein IWS1. And you'll see really quickly that it's acidic. So that's one of the things I talked about, that these acidic ones that you very often get a high number of modification sites. But this has 3 different phosphorylatable residues. And so if you look at this, then all 3 of these versions of the modification would be chemically identical from a mass to charge ratio, where you have the phosphates in a slightly different location. And also, when you separate these chromatographically, they are going to travel through the columns together because they're chemically identical. So there's nothing in here that's going to be able to distinguish them from each other. So when you put them into the mass spectrometer, they're going to come through as a mixture if they all 3 do exist in the same biological sample. And so then that causes this to be problematic and can lead to an ambiguous site localization without clear diagnostic fragment ions in MS2 or MS3. This is also going to lead to a poor delta CN score. And so that's something that you should consider if you're doing a false discovery, if you're doing a PTM analysis, that you may want to adjust the delta CN settings. Okay, so then that's all that I have today for the SEQUEST section. So if anyone has any questions, feel free to ask so that we can discuss it at the Q&A on Friday. And thank you all for your time today.